next we have Kasturi Rajashekar, AGM Business Development at Ricky Global Trading, and he will be speaking about unveiling new chiral chemistries to address industry challenges.
Um, Good afternoon, everyone, and welcome you all for our session on unveiling new chiral chemistries to address industry challenges. My name is Rajshekar Kasturi, working as an AGM Business Development at Ricky Global Hyderabad. So today I will take you through this agenda. So I'm going to explain all these things using case study. That case study explains the purification challenges, how we are going to facing the challenges every day, and then that going to be solve few of the chiral chemistries which is offered by the resist technologies. During this case study, I'm going to discuss about different chiral stationary phases offered by the resist technology, which is the traditional chiral phase technology, which is polysaccharide based, and also we have a different kind of chiral stationary phases, which is prickle type, and also we have a core shell technology that is going to give the special interaction, different interaction with the different type of compounds. And finally, I will be touch upon SFC versus LC, and then I'll conclude this presentation. So, chiral separations are completely different from the achiral separations because the chiral physicochemical properties are both are same. So when the racemic mixture is passing through the stationary, one plane of the stationary phases, the R isomer is going to interact more with the stationary phase because it's going to interact 3-point modal, whereas the S isomer is going to interact 1 or 2-point modal. So that R isomer is going to interact more with the stationary phase and it is going to retain in general, so it is going to elute after this S isomer. So there are a different type of interactions was mentioned here. To explain our features in the chiral chromatography, I am going to take this well-known example naproxen, which contains R isomer and the S isomer. Here, S isomer is a non-steroidal anti-inflammatory drug which is administered as a single enantiomer. And whereas in the R isomer is actually toxic. So it is necessary to purify the S isomer before going to the drug making, so, production. That's why for the target or aim of this study to develop a purification method which should be simple, fast and effective. So, in general, the method development, there are many type of method developments which is following in the industry. But however, the resolution is directly proportional to the selectivity. The selectivity is altered by the stationary phase as well as the mobile phase. So here, in the advanced method development cases, we are going to do the different type of stationary phases which are going to use, and also different type of mobile phases in the screening experimentations. Okay? Once we got the best experiment, we are moving to the optimization stage where we are going to use the other method parameters such as solvent composition, temperature and flow rate. So in our headquarters, we have a system that can operate both LC as well as SFC using the same instrument. We do have a different type of capability over here with a different solvent composition, and also we have a column switching valve which can able to connect the more than 6 columns at once. Because of that, we are going to connect all the columns and the method development will be much faster compared to the traditional instrument. So during this initial method development, so we are going to use the traditional polysaccharide stationary phases which is available with the resist, that is immobilized stationary phases and all coated stationary phases. In this we have injected the naproxen mixer, so we did not get any of the separation using this traditional chiral stationary phases. Then we continued our method development by using the another type of stationary phase, that is Pirkle stationary phases. The importance of these stationary phases which we have, it is covalently bonded with the semi-rigid scaffold, and also it is having a multiple ionization center, multiple interaction centers. You can see is the blue color which is mentioned tetrahydrofuran, which is a pi electron donor, whereas in the green color circle, which is the dinitrobenzoyl, which is going to be having a pi electron acceptor, whereas we have a amide functional group which is be acting as a hydrogen bonding acceptor as well as donor. So because of these multiple interactions, when we are injecting the mixture of the compound, we are going to get much separation compared to the other type of chiral separations. And the beauty of this stationary phase, it is also having a two chiral centers. So we have a chance to, or we have to configure this stationary phase into the two different functional group, that is the RR and SS chiral stationary phases. The importance of this different configuration I am going to talk about in the coming slides. So when we injected our racemic mixture into this chiral stationary phase, we got a good separation, better than the separation which we got in the polysaccharide based stationary phases. You can see we got more than 3 resolution. But when we are increasing the sample concentration for the PrEP HPLC, we got the separation like this. So it is not completely base to base separation. Because of this, we are losing the yield and the purity of the compound. So for that, we continued our method development, and we have gone through some of the literatures, and we found out one of the literature over here, and they were classified different type of stationary phases they were used, and different type of separation mode also they have used. Anyhow, they did not got the more than 3.5 resolution in any of these stationary phases. Then what we have done, we have used our different configuration Welk 1 phases. What is happening, you can see here, in the RR Welk phases, the main peak is eluting at the first, and then small impurity is eluting later. When we change the sample configuration here, the R isomer is eluting first, then the S isomer. So this is the key takeaway from this type of stationary phases. When we injected the higher volume of the sample, so we got the base to base separation. We can see here that clearly from the RR Welk to SS Welk. So this is easy as to collect the both fractions without changing any method development. If you want to try the again method development from the scratch, it will take longer time. Instead, just switching the one column, so we got the base to base separation, and we can get the best purity from this. So we have a study from the comparison between the HPLC and SFC. You can see here this red color indicate the LC, and then green color indicate the SFC. We can see the runtime is 3 times better than the LC when we are using the SFC. So, and also we have a higher loading capacity compared to the LC, SFC having a higher loading capacity, and also it produced the high yield as well. So in this case, SFC is giving the best results compared to the PrEP HPLC. And we, after collecting the different fractions, we have injected into the HPLC, and we calculated the enantiomeric excess for the both impurity, and we got more than 99% for both of the impurities. And then the same stationary phase is also available in core shell technology. We do have a much more advantages when we use the core shell technology. We can also increase the more number of volume and injection as well. But you know the benefit of core shell technology. But here the one claim is it is going to provide the UPLC performance at high HPLC pressures. And also we do have a different type of chiral connectors, and which will be very much useful for the specific applications. And the application was mentioned over here, and also you can go through our website. We were mentioned over the website as well. And then to conclude my presentation, whenever we have a small impurity in the tailing front, So instead of doing the new method development, we can simply switch into our SS to RR stationary phases. Then we will get the base-to-base separation that will be help us to collect the both very high pure impurities as well as the main compound. And also, the same stationary phase can be useful for the SFC technique as well. So as seen in the example, the SFC offers high sample loading capacity as well. And also, the Regis is offering same stationary phase right from UPLC to LC and SFC as well, so that when we are developing a method using the UPLC for fastening the method development and same thing you can apply to the prep HPLC as well. And we have a special type of core shell technology that we seen in here that will be applicable for the special problems. Okay, and I would like to thank you everyone, and please go through our website. There are a lot of literature, articles, posters available over there. We can go through it. Thank you.
Thank you.